After studying this module, you shall be able to know about the significance of toxicokinetics, the various stages of a typical toxicokinetic process, the concept of toxicodynamics. The explanation of the pharmacokinetics or toxicokinetics involved in absorption, distribution and elimination processes is a highly specialized branch of toxicology. Toxicokinetics is the quantitative study of the movement of an exogenous chemical from its entry into the body through its distribution to organs and tissues via the blood circulation and to its final disposition by the way of biotransformation and excretion. Toxicokinetics represents extension of kinetic principles to the study of toxicology and encompasses applications ranging from the study of adverse drug effects to investigations on how disposition kinetics of exogenous chemicals derived from either natural or environmental sources or xenobiotics govern their deleterious effects on organisms including humans. The elementary kinetic concepts for the absorption, distribution, metabolism and excretion of chemicals in the body system initially came from the study of drug actions or pharmacology. Toxicokinetics is defined as the rates of all metabolic processes related to the expression of toxicological endpoints. The human body can be open to a range of toxicants that may be existent in different environmental media such as air, soil, water or food. However, just merely being exposed to these dangerous chemicals does not certainly transform into a toxicological response, as the toxicological response is often associated to the exposed dose, relations between the toxicant and the body's barriers and defense mechanisms will have an effect on toxicant movement in the body and eventually control the degree and magnitude of toxicant absorption and distribution to the target tissue. The overall entry depends on both the amount present and the saturability of the transport processes involved. Liver metabolism will have the most significant effect on toxicant bioavailability following gastrointestinal absorption, but microbial activity and various enzymes in the gastrointestinal tract and the skin play a significant role in oral and dermal absorption respectively. Physiochemical features of the toxicant such as the chemical form can be a valuable indicator of whether the toxicant will be absorbed and distributed in the body. The understanding and interpretation of toxicological findings require information on two key areas. Firstly, delivery of the compound to its site of action, that is toxicokinetics, and secondly, the mechanism of action and potency of the chemical at the site of action, that is toxicodynamics. First, the significance of toxicokinetics. Toxicokinetics is an extension of pharmacokinetics in that these studies are conducted at higher doses than pharmacokinetic studies, and the principles of pharmacokinetics are applied to xenobiotics. In addition, these studies are essential to provide information on the fate of the xenobiotic following exposure by a defined route. This information is essential if one is to adequately interpret the dose-response relationship in the risk assessment process. The principles of pharmacokinetics apply to any environmental, xenobiotic, and many of the biological processes involved in the absorption, distribution, and elimination, such as the metabolizing enzymes, are non-specific and shared by drugs and other low molecular weight organic molecules, such as additives, pesticides, and contaminants. Toxicokinetic data have the potential to define the internal exposure in animals based on plasma or blood concentrations of the parent compound or its active metabolite in relation to the dose given to the animals. The relationship between plasma or blood concentrations and those at the site of toxicity. The information allowing quantitative interspecies comparisons derived from appropriate blood or plasma data after the administration of tracer doses to human volunteers. The term toxicokinetics is now widely accepted to describe pharmacokinetic processes for potentially toxic chemicals which do not have a therapeutic effect. Toxicokinetics should also be considered to confirm appropriate exposure levels in different treatment groups. Toxicokinetic process includes following phases. First is absorption. Absorption is the acceptance of a constituent from the surroundings into the organism. The mammalian body has a number of characteristic defense mechanisms and membrane barriers that have a tendency to check the entry or absorption and distribution of these toxicants once an exposure event has occurred. However, if the toxicant is freely absorbed into the body, there are still other anatomical and physiological barriers that may prevent distribution to the target tissue to elicit a toxic response. In order for a xenobiotic to reach its site of action, it must pass across various body membranes, that is, cells of skin, cells of lung, gastrointestinal tract, erythrocyte membrane, etc. As soon as the toxicant has been absorbed, the toxicant molecules can move around the body in two ways: by bulk flow transfer, that is in the bloodstream, and by diffusional transfer, that is molecule by molecule over short distances. There are following types of absorptions. First is pulmonary absorption. The lungs are the primary route of deposition and absorption of small airborne particles, gases, vapors, and aerosols. For extremely water-soluble gases and vapors, a substantial part of the uptake ensues in the nasal cavity and the respiratory branch. But for less soluble substances, it principally takes place in the lung alveoli. Second is percutaneous absorption. The skin is a very efficient barrier. Apart from its thermoregulatory role, it is designed to protect the organism from microorganisms, ultraviolet radiation, and other deleterious agents, and also against unnecessary water loss. Next is gastrointestinal absorption. It occurs after accidental or intentional ingestion. Larger particles initially inhaled and deposited in the respiratory tract may be absorbed after mucociliary transport to the pharynx. Almost all soluble substances are well absorbed in the gastrointestinal tract. The low pH of the gut may expedite absorption, for example, of metals. Other routes in toxicity testing and other experiments, exceptional routes of administration are often used for accessibility. Although these are uncommon and ordinarily unrelated in the occupational setting. These routes include intravenous, subcutaneous, intraperitoneal, and intramuscular injections. Overall, substances are absorbed at a higher rate and more absolutely by these routes, particularly after intravenous injection. This leads to short-lasting but high concentration peaks that may increase the toxicity of a dose. Disposition is the term often used to describe the simultaneous effects of distribution and elimination processes subsequent to absorption. Second is distribution. Absorption of toxicants into the blood needs to be high enough so that it will have a significant effect at the site of action in other areas of the body. The distribution process that takes the absorb drug to other tissues is dependent on various physiological factors and physiochemical properties of the drug. This process is therefore a reversible movement of the toxicant between blood and tissues or between extracellular and intracellular compartments. There are, however, several complicating factors that can influence the distribution of a toxicant. These include lipid solubility, pKa, and molecular weight. For many toxicants, distribution from the blood to tissues is by simple diffusion down a concentration gradient. The concentration gradient will be influenced by the partition coefficient, or rather the ratio of toxicant concentrations in blood and tissue. Tissue mass and blood flow will also have a significant effect on distribution. For example, a large muscle mass can result in increased distribution to muscle, while limited blood flow to fat or bone tissue can limit distribution. There are unique anatomical barriers that can limit distribution of toxicants. A classic example of such a unique barrier is the blood-brain barrier, which can limit the distribution of toxicants into the central nervous system and cerebrospinal fluid. Generally, after a toxicant or drug is absorbed, it can be distributed into different physiologic fluid compartments. The obvious volume of distribution, that is VD, is defined as the volume of fluid necessary to contain the total amount A of drug in the body at the equivalent concentration as that present in plasma Cp. Expression shows Vd is equal to A by Cp. Next is biotransformation. Vertebrates have advanced the ability to convert many toxicants into other chemicals, every so often detoxifying the chemical and sometimes activating it. The same or similar biochemical processes that hydrolyze, oxidize, and reduce toxicants in vertebrates also take place in many lower organisms. Biotransformation is the process which leads to a metabolic transformation of foreign compounds, xenobiotics, in the body. The process is often mentioned as metabolism of xenobiotics. As a common rule, metabolism converts lipid-soluble xenobiotics to large water-soluble metabolites that can be efficiently excreted. The liver is the foremost place of biotransformation. Xenobiotics, once inside the body, undergo a series of biotransformations. Biotransformations follow a complex series of chemical reactions that are enzymatically facilitated and are ordinarily irreversible reactions that are actively positive, resulting in a reduction in the Gibbs free energy of the system. Thus, the potential for biotransformation of a chemical depends on the reduction in free energy that results from responding of the chemical with other chemicals in its surroundings. Those reactions that introduce a new functional group into the molecule by oxidation, reduction, or hydrolysis are designated phase 1 reactions, whereas the conjugation reactions by which phase 1 metabolites are combined with endogenous substrates in the body are denoted as phase 2 reactions. Chemicals may go through any one of these reactions or any combination of them, either concurrently or subsequently. Because biotransformations are catalyzed by a large number of enzymes, it is to be expected that they will vary among species. Qualitative differences imply the occurrence of different enzymes, whereas quantitative differences imply variations in the rate of biotransformation along a common metabolic pathway. The differences resulting from variances in enzyme levels, in the degree of competing reactions, or in the competence of enzymes capable of reversing the reaction. Most biotransformations are detoxification reactions. Many oxidative reactions produce reactive metabolites. Next is elimination. The ability to efficiently eliminate toxic materials is critical to to the survival of a species. The complication of toxicant elimination processes has increased equal with the increased complication associated with animal form. For unicellular organisms, passive diffusion can suffice for the elimination of toxic metabolic wastes produced by the organism. Three main routes of elimination culminate in the specialized organs of elimination: the liver, kidneys, and the lungs. The liver assists as a major organ at which lipophilic materials are collected from the blood, biotransformed to generally less toxic and more polar derivatives, and then eliminated into the bile. The kidneys balance the liver in that these organs accumulate wastes and other chemicals in the blood through a filtration process and eliminate these wastes in the urine. The respiratory membranes of the lungs are perfect for the removal of volatile materials from the blood into expired air. Next is renal elimination. The kidneys are the spots of elimination of water-soluble chemicals that are removed from the blood by the process of reverse filtration. Two characteristics are principally responsible for determining whether a chemical will be eliminated by the kidneys, or that is size and water solubility. Next is hepatic elimination. The liver assists many vital purposes to the body. It has a large volume to hold blood and thus helps as a blood storage site. The liver produces and secretes many constituents that are required for typical biological function. It purifies the blood from several endogenous and foreign molecules. It biotransforms equally endogenous and exogenous materials, decreasing their bioreactivity and assembling them from removal. It eliminates wastes and foreign chemicals through biliary excretion. Three of these functions occur coordinately in a method that makes the liver a chief organ of chemical elimination: chemical uptake from blood, chemical transformation, and biliary elimination of chemicals. Next is respiratory elimination. The lungs are extremely specific organs that function in the uptake and elimination of volatile materials, that is, gases. Consequently, the lungs can work as a major site for the elimination of chemicals that have a high vapor pressure. The functional unit of the lung is the alveolus. These small, highly vascularized membranous sacs function to exchange oxygen from the air to the blood uptake and equally exchange carbon dioxide from the blood to the air, that is elimination. This interchange occurs through passive diffusion. Chemicals that are adequately volatile may diffuse across the alveolar membrane, resulting in removal of the chemical from the blood and elimination into the air. In addition to these major routes of elimination, several quantitatively minor routes exist through which toxic materials can be eliminated from the body. These include skin, mammary glands, hair, and sweat. Next is bioavailability. Bioavailability, the fraction or sometimes percentage of the administered dose that enters the general circulation as the parent compound. In the absence of pre-systemic clearance of first-pass metabolism, the fraction is 1. In oral exposure, pre-systemic clearance may be due to metabolism within the gastrointestinal content, gut wall, or liver. A low bioavailability may be due to poor absorption or first-order conditions, and the state of being capable of being absorbed and available to interact with the metabolic processes of an organism. Bioavailability is characteristically a function of chemical properties, physical state of the material to which an organism is exposed, and the capability of the individual organism to physiologically take up the chemical. First-pass metabolism will reduce the systemic absorption of the substance and instead increase the absorption of metabolites. This may lead to a diverse toxicity configuration. The bioavailability must be understood with the purpose of determining what dose will prompt the anticipated therapeutic effect. It will also clarify why the identical dose may cause a therapeutic effect by one route but a toxic or no effect by another. Next is toxicodynamics. Toxicodynamics is the study of the biochemical and physiological effects of toxicants and regulates their mechanism of action. Toxicodynamics is the process of interaction of chemical substances with target sites and the subsequent reactions leading to adverse effects. It states about the molecular, biochemical and physiological effects of toxicants or their metabolites in biological systems. These effects are consequences of the interaction of the biologically effective dose of the active form of the toxicant with the molecular target. Toxicodynamics can be used in combination with toxicokinetics in environmental risk assessment to determine the potential effects of releasing a toxicant into the environment. We'll conclude this module with the summary. Toxicokinetics is defined as the rates of all metabolic processes related to expression of toxicological endpoints. It is the method of the uptake of possibly toxic substances by the body, the biotransformations they go through, the distribution of the substances and their metabolites in the tissues, and the elimination of the substances and their metabolites from the body. Toxicokinetics should also be considered to confirm appropriate exposure levels in different treatment groups. Under unusual conditions, facts on absorption, distribution, metabolism, and excretion. The absorption, distribution, and elimination of a drug are qualitatively similar in all individuals. However, for a number of reasons, the quantitative aspects may differ considerably. Each person must be considered separately and doses adjusted accordingly. Primary causes of admittance of toxicants to the human body are dermal, gastrointestinal, and respiratory. Extra routes come across in experimental studies contain intraperitoneal, intramuscular, and subcutaneous routes. When direct entry into the circulatory system is preferred, intravenous or intra-arterial injections can be used to bypass the absorption phase. Bioavailability is the fraction of the contacted dose that is relocated from the site of contact or administration into the common circulation or tissues. Toxicodynamics describes the mechanism by which a toxicant interacts with the target site.